Thanks for the introduction, Kiman, and thanks everyone for the invitation. So in particular, Andrew, you helped with the organization, Kiman, David, for the invitation to talk to you. Yeah, I'm very sad that I couldn't make it to Melbourne this year, but yeah, maybe we can fix that later. So yes, it's pretty much work in progress. Okay, so that was my attempt, my journey to see what agent-based models are doing, and realizing that actually I have played with those in the past, but that was not the naming I used at the time. So yes, I will. First of all, I'm going to talk about my background and explain how I came to being interested in agent-based modeling recently. And then I'll try and explain what an agent-based model is and show two applications to agent-based model. Actually, maybe more than two. I'll skip through very quickly and talk about genetic algorithm for a few minutes. So yes, what I do usually, my overall research theme is the study of complex biological systems. And by complex system, I mean a set of many interacting entities, so biological entities, that somehow show a common behavior. So say if you study, and I'm going to talk about that very quickly, if you study gene networks, you have lots of variables and you arrange them into a graph or a network, and you have edges that show interactions between them. And in the end, what happens is that the organism is showing some metabolism or some behavior response to the environment, for example. And that's exactly what we try to see, this kind of emerging behavior of the system. So more precisely, lots of my work so far was focused on reconstructing networks from high throughput data. So yes, if we look at the modeling that looks like this, think of that as a map of the functioning of an organism, a living organism, where you have the gene space, protein space, metabolic space. So yes, I'm assuming that everyone here is familiar with very basic molecular biology concepts. If not, raise your hand quickly, and I can try and fix that very quickly. Yes, so the gene space, for example, you see that gene 2 is expressed and translated into a protein. So it's transcribed first into a messenger RNA, translated into protein 2. And protein 2 has a repressive effect on the transcription of gene 3. The translation of gene 3 and gene 4, they interact together to form a complex that would be used as an enzyme in the reaction that transforms metabolite 5 into metabolite 7. And say metabolite 4 would trigger one phenotype of interest. Yeah, okay, so you have a large number of complex relationships. What you can do in systems biology when you gather data is that you can have observations at the different spaces. Sometimes, if you're lucky and if you're rich, you can have observations in more than one space, actually. And you can try to align them. And the goal for me is to reconstruct the edges you see. So either the plane edges that are physical relationships and in the functioning of the organism, or the dashed edges, which are indirect relationships. Yes, so then that means that technically I'm still a mathematician or a statistician. That means I'm interested in probabilistic graphical modeling. That means I use Bayesian networks or Markov random fields to jointly model observations and a non-independent framework. But what I mean by that is that I use a graph. Okay, so the variables are not independent. So I hit the high dimensional setting. I have a large number of variables usually on a small number of samples because we have a limited number of experiments to gather data. The data can be heterogeneous and noisy. So, for example, you would be accustomed to Keyman's work, who tries to integrate data from different sources, so from different labs. We can encounter missing data. Either you want to impute them or you want to study the effect of having missing data on the quality of the reconstruction of the graph you're doing. So that you can have missing data in one layer, even if you have observed data in this layer, but you can also have missing data in an entire layer. And what it means in terms of the reconstruction of the interactions usually means that you're going to reconstruct indirect interactions. And how do you interpret those? Recently, so recently means in the last 5 years, I've also been interested in putting the causal label on the relationships we infer in the systems. So which causal relationships can you infer and which you can't? And say an interesting question would be, what is the minimal set of interventions you can dream of in your data set to clear all ambiguity in the causal graph? Yes, and then I start to have a good list of applications with colleagues, either I had in Toulouse, at Massey, across New Zealand, or a monogenic diseases, for example, would be with colleagues in Marseille as well. Yes, so now, I was going to say last month, but actually that's February. I was lucky enough to spend 3 weeks in Marseille at the CIAM at a conference that was a series of workshops, and I went to 3 of those. So week number 3 was devoted to the study of infectious diseases and the modeling of those. The week number 4 was very attractive to me, but actually I was quite surprised by the content. It was a week devoted to complex systems in biology and medicine. And week number 5, well, maybe I would advise you to look at the videos online on the CIAM website. You would see a very good lecture of Keyman, who talked to us about a modeling of molecular networks. So yes, going to talk very briefly about one talk. Was quite impressed, maybe because that was one of the talks where I wasn't asleep. It was on a modeling through partial differential equations of foraging ants. Yes, and then the first week, I was quite surprised to see people, modelers, epidemiologists who are very passionate. So that didn't mean I didn't join them for the night sessions where they were coding like crazy because the COVID was just out and they were super interested. But what surprised me is that most of the approaches I saw in the talks, I'm not talking about week number 5, but for the week number 3 and 4, study of modeling in infectious diseases and the modeling of complex systems in biology and medicine, was that very few of them would use probabilistic simulations or even probabilistic reasoning. The stochasticity would be in the noise they put on the equations they use to model the system. So yes, another motivation was that I was to start a disease outbreak challenge on African swine fever. So it's a mean disease for pigs that happens in farms through the wildlife. And then the goal was to predict optimal control strategies on an island to try and stop the spread of the disease, or actually, well, do our best. So the challenge has been postponed because of the COVID situation, but it's due to start soon, I hope. And yeah, I have a student waiting for that, for their data. And I've also been thrown in that by a small consulting I'm doing with the NDHB, where people wanted to assess the number of beds required in hospitals in response to the COVID outbreak here in New Zealand. Was there any question? Nope. Okay. Sorry, thought I heard someone. So yes, I'm going back to the modeling of ant. So that's the chased on the slide that Sean Ryan has been using. And these 4 plots, so plot number A on the top left shows ants, so that's the pink dots that are close to their nest on the left, and on the right, that's the food source. So at first, what happens is that ants would be roaming freely everywhere in a random walk. And in B, the first ants start to locate the source of food. So what happens, maybe you see it, a few ants did harvest some food and they're coming back towards the nest. And the most interesting plot for me is plot number C, where you see the trails of ants that go back and forth from the nest to the food source. And the important failures, or that's what Sean said, and presumably it seems to be a realistic behavior, is that the number of trails which is formed by the ants going to the source of food or coming back seems to be odd. So you don't have one trail of ants going to the source of food and one trail coming back. It's either 1, 3, 5, or 7. And he clearly showed that in the simulations he presented. So yeah, and then the plot on the bottom right as the food source as over, and then the ants are roaming freely again to try and gather another source of food. Another failure he was interested in, but he didn't observe that through his set of equations, was branching processes in the case where you have several sources of food present in your space. So what he has in his simulation is that the ants would exploit one source and then they would switch to another one. So basically they start with the closest one. So then I was sitting in the lecture hall and I thought, wow, I'm pretty sure that I could implement that easily. And that was a big mistake from my part, by doing some simulations on my computer. So a few words on agent-based models first. The model is going to be useful if it's a description of a process, and that means that I want to describe the world in terms of the objects that are in this world and the events that occur. Obviously, it's the modeling exercise stresses some aspects of the real world and it totally forgets the rest or simplifies many of the aspects. So in this model, I want to have agents which move autonomously. They are entities with inherent properties and they can interact with each other or they can interact with the environment. So I need to define their position and their status. So their status could be whether the ant senses a chemical trail, or it could be whether it's carrying food. It could be whether another ant is too close to her on the ground. So agent-based modeling conveys the idea that a process can be described through these agents, the environment, and a simple set of rules. Okay, when I say simple, that means it's a few rules. If you take a piece of paper and you start writing them down, that's easy. It boils down to 3, 4 rules overall. And the goal is to have an interpretable but still quantitative view of the world. It's not to make precise predictions of what is going to happen. So it's not going to give me predictions of the source food is going to be exploited from day 2 to day 8, and then they switch to another one. I wouldn't see that. I would see general behavior emerging from the interactions of the ants. And what you want to look as well as the sensitivity to the initial conditions. So would you have a robust system or would you be close to chaotic behavior? And that's what leverage points are. So you can identify points, say, for example, in the parameter space, where you would have a drastic change in the global behavior you observe from the interactions of your agents. When you do a small change, a relatively small change in the parameters. And hopefully I'm going to show that to you through a few examples. So yeah, the representation of the agent-based model is key. You don't want to be trying to write all the individual equations. Say if you have a flock of birds, and we all know that they have this behavior where they stay together, but when a danger approaches, they all go around the danger. So they avoid it. You can't imagine trying to write all the equations that explain why the birds stick together, then why suddenly once they become a prey, they would go away and come back together. Agent-based model would be, they stand in the middle of the path between having inductive reasoning, where you start from data and then you build theory. That's what statistics would do usually. You take a data set, you analyze it, and then try to make sense of the relationship you see in your model. So you have a list of p-values in your coefficient in a regression, and you try to make sense to interpret those p-values and the strength of the interaction, not through p-values. And the other way would be to have a deductive reasoning, where you start from first principles and you try and build a more complex theory. So the first principles would be, you know how your ends interact, then can you predict the number of trails they are going to form when they go to the food source or when they come back? So what we do, we are in the middle, what we do is we generate the observations from first principles and we observe them, we observe it from simulated data. Conversely, if the pattern is known, what you can do, you can try and build these first principles, these rules to create that pattern. Yeah, so agent-based models are well suited when you have a medium-sized system, simply because if the system is too small, you don't have enough agents to create the observed pattern. And where if you have too many agents, sometimes you don't see the pattern as well, or actually you have a big computational issue. They are very efficient when you have an heterogeneity of the agents. So all the agents don't need to be the same. That'd be if you have a system where you have sheep and wolves, for example. Whereas it would be quite hard to try and model that through equations. Actually, 2 stages is okay. More than that is going to be complex. You can model complex nonlinear local interactions between the agents or with the environment. And very often in agent-based models, the space plays a role, and you have spatial-temporal relationships as well. You can also have an evolution or an adaptation of the agent to the environment. So you use the history of the agents to adapt their rules, their behavior. Yeah, so I've briefly talked about the comparison between agent-based modeling and equation-based modeling. Equation-based, usually you would apply that to a continuous space, and you have this homogeneity assumption. You also need to have the aggregate knowledge. So you put that, it's an input of your modeling. So you would say, and that's what Sean was doing with his hands. He was saying how the ants, he knew the ants would avoid each other. He knew that so the equations they were not directly modeling the fact that there were an odd number of trails or that there was branching, but it was more than a simple description of what an ant would do locally. Whereas the agent-based model is more bottom-up. But yeah, the 2 approaches complement each other, I think. So very quickly, limitations. We've talked about the computational limitations. Lots of parameters, so that was an issue for me. I ended up with a list of, so with the ants, 5 or 6 parameters. I had no clue how to set them. So I read a lot about that. With the epidemics modeling, I had much more than that. You need some knowledge on the individual behavior. So again, you need to create that if you're not an expert. It's hard to see a cause to effect pattern as well. I remember one of my professors at the university was comparing, he said that physics was the thrown science. You could do an experiment, you could modify one factor and you could see the effect. Chemistry, you're staying on a stool. It's very hard to have one factor that you can set individually. Biology, it's too messy. So even if you think you touch one factor, actually you can have many different causes for the observed consequence. So yes, with agent-based modeling, you need to have this back and forth movement between real-world experiments to make scenarios plausible and then suggest experiments as well. What I noticed as well is what I would call the lack of statistical control. So how do you validate your model in the end, or how do you validate your parameterization? So think about what's the real dimension of my space. So I think with the COVID modeling, I end up with a list of almost 50 parameters in the most complex model I have. And say, when do you see that two models are equivalent? Something I haven't come up with yet. Yes, so going back to foraging ants, very quickly, I said that ants would look for food randomly. When they locate the food source, they go towards it. And well, when they locate the food source, it means they are at the food source and then bring the food back to the nest. By doing that, they leave a trail of pheromones, that's chemical compounds on the floor, and the ants can detect it. And actually they follow the gradient of this chemical trail. And the more ants are on the track, the stronger the trail. Okay, so what happens is that these pheromones, they diffuse so that the ants in the vicinity of the trail can detect it, but it also evaporates. So that's the two controls I have. Yeah, so that's the first movie. So I'm going to try and launch that. So that's the first simulation I'm going to show you, and I'm going to show you two simulations. This one has got a high diffusion of, stop sharing this, and I'm going to share this one. Okay, so the ants start from the nest in the center, they roam freely, and then they find the first food source. Actually, they found two of them. I put them too close. You see a first trail that forms on the right. They go back and forth. They're supposed to change color when they come back. They should be red. And what I was hoping is that I could start seeing branches. Yes, so you see them from time to time. They follow the same trail. What I didn't see is that I didn't see this number of trails that were supposed to be formed. So either I was hoping to see three of them maybe, so one trail of ants going to the food source and then two trails on the sides. Maybe because the number of agents I have is not enough, or they're too big, because remember that they should avoid each other. So that was with a high diffusion and a large evaporation rate. Now, if I can, I'm going to show you, yep, the second simulations, where this time, where's Zoom? Okay, I have a smaller diffusion rate, but the evaporation rate is smaller as well. So the pheromone stays on the ground for longer. And actually that's what you see, that the trails form very quickly. And yeah, there is clearly a branching process between the two food sources on the left, but again, I didn't see any trail appearing. or nothing obvious. Okay, should go back to close this and go back to my slides. Yep. So yes, a small discussion of what I observed, and you might jump at me as mathematicians or statisticians and say, well, what have you shown here? You just showed simulations and your results. I don't know how to formalize that. So each ant follows simple rules. And I kind of saw a global behavior of the colony that emerged. Oh, that's typo number 7 at least. I saw that it was hard to form a stable trails to more distant forces, all the more that when there is a food source which is close, then the other ones are neglected. And that makes sense, but because you have less ant workers available to go and gather food that is further away. I also noted that you, and I didn't show that, but that you need a critical number of workers to exploit distant food efficiently. Otherwise, well, the trail doesn't stick for long enough. I haven't implemented any strategies to find the way back to the nest. So what I did is that once the ant had food source, had found the food source and had harvested food, it was coming back to the nest. It knew how to come back to the nest. I'm not too sure that the pheromone gradient is the only strategy that the ants use, either to go to the food source or actually to come back, because I think that's what they do. They follow these trails as well. I've not been able to really reproduce the trails of tracking and hoarding ants. And I didn't see the odd numbers of trails observed in nature or in the work by Sean Ryan. Yes, and then so then I'm thinking maybe there exists data on that, but how would I fit data to my simulations? I'll discuss slightly more on that later. I'll probably skip this part, or actually, that was when I was preparing this talk, I realized that I had some experience with agent-based models beforehand in the form that I used genetic algorithms. So the first genetic algorithm I used was with the work of a PhD student, Magali Champion, in Toulouse. So it started, I think, more than 6 years ago. And we were interested in the reconstruction of directed acyclic graphs from gene expression data. So we were using a combination of convex programs and a tailored genetic algorithm. And yeah, if you want to try the R package, the GetAg R package, I saw that there is a new version that's just been out. And the other application I've been using with genetic algorithm very recently was with colleagues in Marseille about the detection of active modules, active in the sense that these are subnetworks of a global multiplex network. So multiplex means there are several layers, which are densely connected and which show differential expression, for example, at the same time. Okay, so yeah, if the paper has just been out on bioRxiv, so feel free to have a look at the paper, and if you want to give us feedback, that'd be very welcome. So the first example, if I talk about it very quickly, you have a gene expression matrix X and a graph that you encode in an adjacency matrix G. For a directed acyclic graph, you can write the adjacency matrix as the product of a permutation matrix P times a lower triangular matrix. So think of P as a node ordering. So in a directed acyclic graph, you have a source and a sink. That means you have a natural order of the nodes, okay? And all the nodes in between would receive a number of connections and give a number of outgoing connections. The T, the lower triangular matrix, contains the quantification of the regulatory relationships. So the strength of the coefficient of the linear equation I just wrote, X equal X, G plus epsilon. And you obviously want to avoid to have anything on the diagonal, otherwise you have a trivial solution. So the idea is that to find the optimal graph or the optimal directed acyclic graph, you need to find the optimal G, and that means you need to find the optimal permutation matrix P and triangular matrix T. So if the permutation is fixed, you can do a classical gradient descent because the fitness function we build is convex and differentiable. On the other hand, if T is fixed, P is a discrete matrix. As a matrix of permutations, that means it's a matrix of zeros and ones, and I think it's the sum of the columns that need to sum to 1. And even for moderate network sizes, you can't enumerate all the possible permutations of the node. So we chose to resort to a genetic algorithm to do that. That means you have a population of networks and we cross them. So each time in these populations, we pick up 2 parents, we cross them to create a child. We mutate them to do random exploration, and we select the best solutions through a fitness function towards the end. So yes, I insist we have several solutions at each step of the algorithm, and we don't try to find the best solution overall. We keep the population of having several solutions, and we do some kind of an average of the best solutions towards the end. And yes, we know how to monitor the convergence of the algorithm. Yes, so that's very quickly, that's the results we obtain. So the data we picked up was an in silico network reconstruction challenge with networks of size 100, and the sample size was 100. So it's not really the high dimensional case, it's moderately high low dimensional. The networks were supposed to be plausible, and yes, the data sets were simulated through stochastic differential equations that mimic chemical reactions, but we didn't know that. We didn't know how the data was simulated. And on the right, you have precision recall curves, 5 of them for the 5 network in the challenge. So precision on the y-axis, recall on the x-axis. So the ideal method would have a precision of 1. That means that all the prediction it does are correct. So when you start having false positive, you lower your precision, and a recall of 1 as well. So you want to move on the right on the x-axis. The recall is the proportion of edges you predict among all the edges that you want to predict. So the ideal method should start at the top of the graph with precision 1 and recall 0, and then stays high and go towards precision 1, recall 1. So we have a set of different methods which are implemented. Random forest is the Gini 3 method that won the challenge at the time. So it's always easy once the challenge is over to overperform the best performing method at the time. And we also implemented a bootstrap lasso, and we saw that, well, the black curve is our curve. So we did slightly better in some networks. Yes, so that was a successful application of genetic algorithms. I'm going to skip this. So yeah, that's the second application of the detection of active modules, and we're very happy because we have a collaboration with biologists, and they found out that the modules we spotted made sense for them. And yeah, what happened with our approach compared to competing methods is that we are able to detect large modules in the sense that they have more than 10 to 12 genes, which was very useful. Yes, and then since it's a seminar about the COVID-19 mini-series in Melbourne, and I promised I would talk about my experience in modeling epidemics as well. It started with a huge amount of reading on my side, and a new kind for me. I read lots of reports, preprints, lots of them. That's the big change. But I also read interesting information on blogs. I followed quite a few seminars, or even I watched videos. So I quote a few of them here. there are a lot around, and those who are interested, I have a huge list of references if you want. By the way, they are clickable links in my presentation if you want. And I totally advise them. My attempts are very poor compared to what some people are doing. So that's the model I ended up using. The model has got birth and death. So the deaths are on the left-hand side. You have natural death. You can die from natural causes, whoever you are, and you can die from coronavirus as well. So the entire population starts in the susceptible state. So that's the S at the top. And the darker the color, the more severe your condition. Then if you are in contact with one of the infectious person in the population, you can become exposed. That's the E state. And then you evolve towards becoming infectious asymptomatic, IA, or symptomatic, IS. And then you may need to go to the hospital, either in the ward or in the ICU if you're in a critical state, that the HW and HC states. And what happens when you're infectious, either you can recover, so that's the R node on the right, or you die. And when you recover, what I included in my modeling is that you could become susceptible again. So for all these stages in the evolution of the epidemics, I need to set probabilities and evolution time as well. So that's where my huge list of parameters appeared. I hesitated for some time between agent-based models and actually a stochastic mass kinetics, but it wasn't clear to me how I could write all the chemical reactions that were going to encode all the relationships I wanted to put. So I haven't been down this path, but that'd be very interesting to try and solve the Gillespie algorithm. And I would also have an issue with scalability. We did that with systems biology recently, but we couldn't go for more than 20 nodes so far. So I had 3 iterations of the COVID modeling I presented. So same compartments, but in the first model, and that's the only model I'm going to show you simulations on, I had random movements and one class of individuals. So that means that everyone moves the same way and has got the same contact with everyone. In the second iteration, I replaced the random movements and I had households and social locations. So I had a school, a supermarket, a hospital, and a pub I couldn't resist. In the third iteration, I also added age and professional categories. So I had infants, school kids, health workers, workers with many of your contacts, retired, and I had different contacts and probabilities of transition. And I also had more realistic compartment durations. So I didn't go too complex. I went for exponential distributions in each of the compartments. Whereas beforehand, all I was doing in the first and second iterations, I was picking out at random at each iteration, so each hour, whether I would stay in my present compartment or whether I would move to another compartment. Yep, so that's the first simulation. So you should see that. So the time as the number of hours. Yep, you should see that moving. So towards on the right, you should start to see dots which are slightly darker. And when they come close to a susceptible, they can touch each other, and that means that they have a probability of transmitting the infection. So you don't really see it, but I see 5, 6 different infectious people. So I start by seeing one infectious person in the entire graph. And I even start seeing some blue. So blue would be the recovered in my representation. Yes, and that's when the outbreak really started. So we are about we haven't made 10 days yet. So after 2-3 days, that's related to the latent period, the exposed period. So even if you have quite a few people infected, you don't see it in the infection force yet, but that's going to burst out in a few days. So yeah, we are approaching 10 days, and you see that it's spread everywhere. So I have put 1,000 agents in these simulations, and I needed to specify the probability of transition between the different states. So when you start to see quite a large number of dark people, I see some dead people, so they are still around. I didn't remove them from the simulations, but they don't interact with anyone. So yeah, maybe I can stop here. And I can show you, oops. Yeah. Yes. So the that would be the counts in the different compartments. So the light green, you see that was at the top towards the start of the simulations. So I simulated that for 6 months. Would be the susceptible, and the well, the darker the worse. So the exposed would be slightly darker, and you see that the first curve that starts towards the first few days. And then the blue curve is the curve of recovered. So I clearly exaggerated the number of dead people in the simulations. That's the black curve you see. So you top up towards the end, you reach 200 dead people through coronavirus in a population of 1,000 after 6 months. That if you do nothing. What was very interesting, so I need to work out on the parameters. I haven't done anything related to data parameters apart from doing something that wasn't totally silly, but that's close. What was very interesting was that you see this second wave that occurs because recovered people reach a peak. So I think the first infection waves, you get 70% of the population that is infected. And then the recovered population starts dropping down, and that means that the susceptible population goes up. And you see a small wave that happens towards day number 100. And actually, I also plotted the effective reproductive number. That's the mean number of infection per infectious person in the population. So yeah, that's a stochastic simulation. So this number can change a lot. I did plot that on the log scale. That's the bottom plot. And you see that the way I compute it, I'm not too sure why it goes above 1 between day 50 and 100, but clearly it goes up again. So when the number of susceptible grows, that's when the infection occurs again. And that's when you have this effective reproductive number that goes higher than 1, and that means that the epidemics goes into an exponential phase again. So yeah, that's a visualization of the entire population where everyone starts susceptible, and you have these phases where they grow exposed and then infected, or when they need to go to the hospital, and then they recover, and they can get susceptible again later along the way. Very quickly, I implemented the effect of having a quarantine. So the graph on the top is the same counts in different compartments. And the graph at the bottom, at day 15, I implemented a strong quarantine and social effect. That means that I reduced the distance where you can have contact with people, and I reduced the probability of an infection when the contacts happen. So say if you wear a mask or you wash your hand, much better. And well, that just kills the epidemics. So that's what you expect. The number of susceptible stops to drop almost immediately, and the number of recovered goes down as well. It doesn't picks up again. So that means there is no further infection after day 15. So yeah, I'm going to conclude on those. I have a model that simulates the counts. So I insist on simulation in what I would call epidemiologically meaningful compartments. And I can use the reproductive number to monitor the spread. Setting the parameters was an issue, and that's what's going to come next for me. So the first experiment, what they were showing, I didn't show all of them to you, that the outbreak was very sensitive to the number of interactions. If you want, that's what we were what was called social distancing, the probability of infection, if there is a contact, and the duration of the illness as well. Many of my simulations would show that the infection would die because I seeded the infection with one infectious person only. What I also noticed, I implemented breaches in social distancing, and that's a nail in the coffin. Even if 10% only of the people do not respect the physical distancing or social distancing that is imposed, then it has almost no effect towards the end of the simulations on the number of infected people. It just slows down the progression. So the slowdown comes from the people who do the social distancing. But those who don't, they keep spreading the epidemics. So yeah, my prospect, I still want to work on that, is to have visits to a central hub. So I haven't shown that to you, but actually that's what I have. And I haven't quantified the effect of these central hubs. I see that the epidemics goes up much more quickly, but I haven't quantified the relative amount of infection in these central hubs compared to being somewhere else, say in the household, for example. I would want to see the effect of testing and tracking. That means that you could take the infectious people when they are symptomatic, and you can put them in quarantine. And actually, I think that's one of the most efficient way of controlling the epidemics. I have also started working on having several communities. So where you would have this plot of people who would be roaming or moving between households. And then say every day you would have a small amount of people allowed to travel between the different communities. And then the effect of travel or the effect of travel ban between these communities. I also haven't implemented, whereas that's what we were interested in at first, the hospital capacity, because that could have a drastic effect on the number of dead people. If the hospital are full and people need to go to the hospital, then it changes the probability of moving from being infectious to going to the hospital to being dead, for example. Yeah, impact of respect of two measures in place. I have talked about that very briefly. Longer term, yes, I want to have these neighboring communities, but that's slightly more complex. What I want to do now is to have the true compartments. So these counts I showed you, they are actually hidden. What we have is partial counts. And I would also account for test accuracy. So that's where I could try and get some data and feed the data, feed parameters to this data and estimate them. So I'm probably going to go for heavy simulations like MCMC. Yes, and then you've probably heard about that. Many of the, we are facing many of known unknowns. We don't know how many people get infected, not infected, number of asymptomatic people, and if they're asymptomatic, not all of them get tested. So that's typically where statisticians could play a big role in that. Yeah, and I'm going to conclude. So thanks for listening to this recent journey in agent-based modeling. I hope you liked it. I have quite some code to share if you're interested. So feel free to contact me if you want. I'm far from it with the ends. So I didn't see the patterns I was hoping to see emerging. I have a flexible epidemiological compartment model with stochastic behavior that can be used, and I'm hoping to use it in many different frameworks. So say, for example, with the African swine fever challenge I want to use. I was very aware of the limitations of the agent-based models in terms that, well, you don't have a model to do it all. You have to build your model for a purpose, but that's something we do usually with the right level of description. And what is slightly disappointing to me is that it seems to be more an art than a science. I also noticed that some factors are very difficult to quantify, especially with humans. So we know that people don't move like drunken people in a 0-1-0-1 square. It was very hard to come up with contact matrices. So people in the UK have very good tools for that. And I brought them the contact matrices they have. But does it represent human behavior? I don't know. And yes, it can be very computationally intensive to do these simulations. So I started coding everything in R, and that's a huge mistake. Another step could be to include economical or other considerations for an optimal decision to help. And yes, the next big step is to integrate the data. So yeah, I have some references, and then thank you for your attention. I hope I wasn't too long. And if you have any question, I would be very happy to take them.
Thanks, Matthieu, and thanks for this talk. I think it raised a wide range of challenges that we face dealing with, simulated data and data. Does anyone has any questions to Matthieu? I might start with one question first, unless there's someone else who wants to ask a question. I have muted everybody, by the way, so you need to unmute yourself. What kind of data real data would you need in order to check your ABM models and their accuracy for COVID-19, say?
I guess I wouldn't be able to check my model as and that's a big issue, the validation. The data I would use as the only data available. So I guess the daily counts and starts to be quite accurate. So I need to feed that to, I don't know what I'm going to do with my hourly simulations. I need to include the proportion of people who are detected or who are counted as compared to the true number of people who have the infection. So I need to have not only those counts, but I also need to have this kind of a large survey of the population to estimate these proportions. I'm not saying put that as an input in my model directly, but have rules that I can update.
Yeah. Elizabeth has a question to you.
Yes.
I've got a question regarding the fitting of the parameters. So do you think right now it's a question of that you're not sure which inference technique to choose, or is it about the data availability that is the problem?
It's probably the data availability. I'm not too sure about the technique because, well, I would be tempted to go for Markov chain Monte Carlo. That's what makes sense to me. And that seems natural to do that. But given the dimension of the parameter space, I have no clue whether I would achieve any convergence at all or any convergence that makes sense. So that'd be a huge amount of work.
Yeah, but would you have to like open up the whole parameter space immediately, or could you say you still let like 80% fixed and only like infer?
Oh yes, no worries. That can be done, but that means that's a scientific process. That means I need to talk to someone who knows what he or she is doing and say, well, do you think that this parameter is reasonable? Can I keep that fixed for now? And then, yeah, update the rest. The most unknown, I guess.
Yeah.
Yeah, but that clearly makes sense.
Okay, does anyone else has a burning question?
I've got one.
Yes, we have a question from Michael.
So I was wondering, are there any formal methods for model checking for these ABM type situations? I haven't seen very much, and there's a quite plausible, but you typically judge it by eye, and I haven't seen anything that goes beyond that.
Okay, thanks, because I was going to answer very quickly and say, well, I think I'm basically ignorant about that. I've been seriously dealing with those only in the last 3-4 months. I don't know, and I don't think there is. That's what I was putting in the limitation of this agent-based modeling under the term statistical control. I don't know. And you have to be careful when you do that, because you could be tempted to build your own function and say, well, I'm happy because I figured out what I expected. So I self-validate what I want to validate. So it's hard, I think. But no, I haven't come across anything.
Okay. Nice. Thank you.
Davis, you have a question?
Yeah, I think that's covered, so I won't hold up people further now.
Okay, any other questions from the floor? I think if not, let's thank Matthieu for his great talk. And just remind you also that the students can stay for half an hour or so to ask more questions to Matthieu. Thank you.
Yeah, exactly. I'll let the Zoom session open, and I might move towards my kitchen to have a tea, but yeah.